There is a fish whose teeth are too long to fit inside its own closed skull, so the skull grew holes to receive them. There is an animal 40 meters long that is not, by any reasonable biological definition, one animal. There is a worm with no mouth, no gut, no digestive system of any kind, that has been eating whale bones through acid it manufactures in its own roots. Roots on a worm, for millions of years while nothing on the surface had any idea it existed. And those are not the strangest things in the deep ocean, not even close. What's strange is the pattern underneath all of it. The deeper you go, the more biology stops looking like something that was designed for a world you recognize, and starts looking like something that was designed for a completely different planet, one that happens to exist directly beneath your feet, right now, tonight, in water so dark and so compressed that the physics of living tissue itself operates under different rules. The deep sea builds animals that should not work. Not in the imprecise way we say that about anything surprising, but in a specific, structural, architectural sense. The proportions are wrong. The features are in positions that make no sense from the outside. The trade-offs that govern every surface animal, between eye size and the metabolic cost of running one, between jaw capacity and the integrity of the skull attached to it, between having a digestive system and not having one, between being opaque and being transparent. Those trade-offs simply don't hold below 1,000 meters. The constraints are replaced. And when you take every assumption that governs surface biology and swap it out for absolute darkness, water at 2 to 4 degrees Celsius, pressure equivalent to a car sitting on every single square inch of your body, and a food supply so thin and unpredictable that starvation isn't a crisis, but a permanent background condition. The animals that come out of that process look like they came out of a different evolutionary history entirely. Because in every meaningful sense, they did. We have been pulling things up from the deep ocean for roughly 150 years. Since the 1870s, when the HMS Challenger expedition dredged up organisms from depths that the scientific consensus of the time considered impossible for life to occupy. The reaction to nearly every new species retrieved from below 2,000 meters has been some version of the same initial response. This cannot be right. Not because the animals are poorly adapted, because they are so specifically, so precisely adapted to conditions we can barely simulate in a laboratory, that their bodies read as impossible when you evaluate them by the standards of everything else biology has produced. The deep sea didn't fail to make normal animals. It decided normal animals were inadequate, and it made something else. Let's begin with what transparency actually means in that world, because it's the adaptation that most immediately dismantles your expectation of what an animal is supposed to look like. The glass squid, Cranchia scabra, is not pale, not translucent in the way a thin piece of paper is translucent. It is transparent in the way a glass of water is transparent. You can see directly through the body wall to the ink sac, the reproductive organs, the pulsing rhythms of the mantle musculature, food moving through the gut, all of it visible without any special equipment, as though the outer layer of the animal simply decided not to exist. In the shallow, sunlit ocean, this would be a survival catastrophe. Every organ exposed, every movement traceable through the body it moved inside. No surface to break up a silhouette, no pattern for a predator's eye to pass over without registering. But in the deep midwater, below the sunlit zone, in the permanent dark where the only light that exists comes in brief bioluminescent pulses from other organisms, transparency means your body returns no reflection, casts almost no shadow, and gives a hunting eye nothing to anchor to. You become, to anything relying on light to find prey, the optical equivalent of empty water. What makes the glass squid's transparency strategy even more architecturally interesting is the flaw built into it. and what the squid does to manage that flaw. The gut is not transparent. Digesting food produces an opaque mass, and there's no biological workaround for that. The chemistry of digestion requires opacity. That dark mass is visible through the clear body wall, and any predator scanning the water for bioluminescent shadows would have a reasonable chance of detecting it. The glass squid's solution isn't biological, it's behavioral. The animal orients itself vertically in the water column, keeping the gut pointed downward, toward the absolute dark below, away from whatever faint light sources exist above. The transparent animal has developed a postural relationship with its own optical camouflage, managing the one visible part of itself by understanding the three-dimensional geometry of where the light in its environment comes from. An invertebrate with no cerebral cortex, an animal we do not credit with anything resembling deliberate thought, has solved a multivariable optical problem with its posture, and maintains that solution continuously throughout its daily existence in the water column. The glass octopus, Vitrellodonella richardi, extends this principle to a body plan that typically has much more visual presence in the water. Octopuses, on the surface, are masters of color change camouflage. They blend into rock, sand, coral, algae, in a real-time performance of pattern matching that uses some of the most sophisticated skin cells in the animal kingdom. The glass octopus abandoned all of that. Its skin has no chromatophores, the pigment cells that allow shallow octopuses to perform their camouflage, and the body is instead transparent, save for the eyes, the optic nerves, and the digestive gland. All three of those unavoidably opaque structures are cylindrical and positioned along the same vertical axis, so that when the octopus is viewed from below, the most common angle of approach for a predator in the mid-water, they appear as a single thin line, rather than the blocky silhouette of an octopus body. The glass octopus didn't just evolve transparency. It organized its unavoidable opacity into the minimum possible cross-sectional profile. The sea salp takes the concept in a different direction. A salp is a barrel-shaped, gelatinous animal that swims by jet propulsion, drawing water through its body and expelling it to move forward. Completely transparent, filter feeding, organized in chains of clonal individuals that can stretch for meters. And the clarity of a salp's body is so total that in photographs taken in good lighting conditions, the salp is visible only by the small internal structures and by the slight optical distortion its body creates in the water around it. The way you can sometimes see a glass sitting on a table, not by the glass itself, but by the bent reflection at its edge. The salp is the bent reflection. The body is the absence between what's around it. In the twilight zone, between 200 and 1,000 meters, there is a technique called counter-illumination that represents one of the most refined camouflage strategies in the ocean, and it relies on the fact that from below, any object in the water column is visible as a dark silhouette against the faint glow of surface light filtering down. That silhouette is the target. That silhouette is what gets an animal eaten, and a remarkable number of deep-sea species have evolved a solution to it that requires active biological computation to maintain. They produce bioluminescent light from their undersides, from photophores arranged across the belly and flanks, tuned in brightness and wavelength to exactly match the ambient downwelling light, at whatever depth they're currently occupying. The silhouette disappears. The predator looking up finds no contrast between the animal and the light above it. The animal is not camouflaged by being invisible. It's camouflaged by generating a precise optical match to its background. The krill do this. Certain lanternfish do this. Some species of squid do this. And the tuning is not fixed. As the animal moves deeper or shallower, as the ambient light level changes, the bioluminescent output from the ventral photophores adjusts accordingly. tracked in real time through light-sensitive receptors that monitor the downwelling illumination and feed that information to the light organs. The animal is running a continuous measurement and output loop, comparing the light coming from above to the light it's emitting below, keeping the two in constant calibration. This is not done consciously. It happens automatically, neurochemically, as an involuntary process as continuous and unreflective, as your heart maintaining its rhythm. An animal the size of a grain of rice is performing real-time photometric counter-illumination calibration every second of its existence in the mid-water, and it doesn't know it's doing it. Below that zone, in water dark enough that counter-illumination becomes unnecessary, the photophore arrays serve different purposes entirely, and this is where the story of deep-sea bioluminescence opens into something that takes time to fully absorb. Roughly 80% of all animals living in the deep midwater produce their own light. 80%. That number, the first time it registers properly, restructures your sense of what the deep ocean looks like in its living state. We think of the deep sea as dark because no sunlight reaches it. That's true of sunlight. The water itself, at depth, is not dark in the way an unlit room is dark. It is lit, constantly, in flashes and pulses and steady glows and sweeping arcs, by the bodies of the animals moving through it. If you could see the entire water column of the Pacific in cross-section right now, tonight, what you would see is not a dark column. You would see a column full of moving light, from the surface to the bottom, produced by hundreds of thousands of organisms simultaneously, in patterns and rhythms and wavelengths that constitute, in the most literal sense, the dominant communication medium of the largest ecosystem on Earth. The anglerfish's lure is the famous example, but the biology behind it reshapes what famous means in this context. The anglerfish doesn't generate the light in the lure, it farms it. The bulb at the end of the modified dorsal spine contains a dense, self-sustaining culture of bioluminescent bacteria, Alivibrio fisheri and related species, that the fish maintains in a specialized organ, providing them with nutrients and oxygen, and in return, they glow continuously. The fish is the housing, the bacteria are the lamp. Some species of anglerfish can modulate the pulse pattern of the lure, varying the rhythm of the bacterial glow, in ways that may mimic the bioluminescent signature of small prey animals, essentially using the lure as a false signal that impersonates the prey's own food source. A lure that tells the prey it's about to be eaten by showing it something that looks like its own dinner, in complete darkness, operated by a bacteria colony living inside a fish's forehead. The dragonfish, Malacosteus niger, operates on a different frequency, in a way that is genuinely operationally clever by any standard. Blue-green bioluminescence is the dominant wavelength in the deep sea because it propagates well in seawater, and because most deep-sea visual systems are calibrated to detect it. Red light doesn't propagate as far, and most deep-sea animals have no photoreceptors capable of registering it, meaning red light at depth is, for the vast majority of organisms, functionally invisible. The dragonfish produces red bioluminescent light from photophores below its eyes, and has evolved red-sensitive visual pigments that no other animal at those depths possesses, meaning it can illuminate its environment in a color that its prey genuinely cannot see, while it watches everything in what amounts to private illumination. Its prey moves through the dragonfish's searchlight without knowing the searchlight exists. The predator is watching in full color. The prey is in the dark. The arms race happening in the bioluminescent spectrum of the deep ocean is being fought at frequencies most participants don't know are in play. The siphonophore arena discovered a third variation on the same principle. Most deep-sea fish are drawn to bioluminescent flickers that resemble small zooplankton, their food source. It's an exploitable reflex, and several predators have learned to exploit it. Arena uses red bioluminescent lures on the tips of its tentacles to attract small fish that, unlike most deep-sea fauna, apparently possess some red light sensitivity. The lures pulse in patterns researchers have described as resembling the swimming motion of small copepods. The fish approaches what it believes is prey, and the siphonophore's tentacles sting and capture it. A colonial organism, something between a single animal and a coordinated society of specialists, figured out how to mimic one species to attract another species that no other predator in the same water column knows how to target. The eyes of deep-sea animals are where evolutionary pressure becomes most visible as a physical force. When the resource being optimized is photons, rare, scattered, arriving from unpredictable directions at intensities approaching the lower threshold of what biological tissue can detect, the organs built to collect them get pushed toward the physical limits of what soft tissue can sustain. The colossal squid's eyes are 35 centimeters in diameter, making them the largest eyes of any animal alive today, or in any fossil record we have. Inside a single eye of that scale, the lens alone approaches the size of an orange. The retinal surface area is larger than most human hands. The optic nerve connecting that eye to the brain is thick enough to be visible without magnification in a dissected specimen. All of that infrastructure exists in service of detecting the bioluminescent signature of prey in water at 1,100 meters, with almost no ambient illumination, from whatever maximum distance the physics of those optics permit, giving the squid the fraction of a second advantage it needs to initiate an attack, before the prey detects the water displacement of an approaching predator and evades. The spookfish, Dolichopteryx longipes, took the optical problem in a direction that shouldn't be possible in a vertebrate. Its eyes don't use lenses, they use mirrors. Concave biological mirrors made of crystalline plates that reflect incoming light onto the retina, forming images through reflection rather than refraction. The way a telescope mirror works, rather than the way a camera lens works. Every other vertebrate eye ever found, in any species, in any era of the fossil record, forms images using a refractive lens. The spookfish is the single known exception. Mirror-based optics have significant advantages at very low light levels, which is presumably why the spookfish evolved them, but the fact that evolution arrived at this particular solution, a solution that physicists understood theoretically for centuries, before anyone found a biological implementation of it, suggests the deep ocean has been running optical engineering experiments for long enough to independently derive solutions from first principles that human engineering took considerable time to work out from the same first principles. The barrel eye, Macropinna microstoma, is worth slowing down on, because the gap between what scientists thought it looked like and what it actually looks like is one of the more instructive stories in the history of deep-sea biology. For decades, the barrel eye was known from collected specimens, and every collected barrel eye looked, in the specimen jar, like a small silver fish with two fixed, upward-pointing tubular eyes mounted behind the face. Researchers described the anatomy accordingly. Papers were written. The barrel eye was classified as a fish with fixed dorsal vision, presumably adapted to scan upward for silhouettes. That description was not wrong. It was just a description of a dead fish. And a dead barrel eye doesn't look like a living one, because the living animal has a transparent, fluid-filled dome enclosing the entire head forward of the brain. A dome that maintains a specific internal pressure and refractive index that acts as a forward lens for both eyes simultaneously, and that collapses completely when the fish is brought to the surface, and the pressure differential disappears. Every specimen ever examined before in situ photography became possible was missing its most distinctive anatomical feature, destroyed by the act of retrieval. Inside that dome, the tubular eyes don't just point upward, they rotate, forward, sideways, upward, tracking anything in the water around the fish through the transparent dome, each rotation guided by muscles attached to the tube walls. A 2004 underwater camera deployment by the Monterey Bay Aquarium Research Institute produced the first footage of a living barrel eye in situ, a small silver fish hovering motionless in the midwater column, inside a perfectly clear dome, with two vivid green tubular eyes slowly rotating within the fluid. The researcher who first reviewed that footage said it took him several viewings before his brain accepted what it was seeing. That response was not a failure of scientific composure. It was an appropriate reaction to something genuinely unprecedented. The opposite of all this optical elaboration is also present in the deep sea, and equally instructive. The benthic animals, the ones living on or near the seafloor in the Hadal zone, below 6,000 meters, in total darkness without even the bioluminescent activity of the midwater, have no use for eyes. The metabolic cost of developing and maintaining a functional eye is real. Blood supply, neural processing infrastructure, the protein structures in the lens, the developmental resources required during embryonic growth. In permanent, absolute darkness, where the probability of encountering any photon worth detecting is essentially zero, the return on that investment drops to nothing. So the investment stops. Over thousands of generations, the selective pressure maintaining eye development weakens and eventually disappears. What remains in some species is a vestigial structure, a small patch where an eye used to be in ancestral forms, still genetically encoded, but no longer properly assembled during development, slowly being reabsorbed by the surrounding tissue. The darkness didn't take the eyes. It simply stopped paying for them, and they went away. Now we get to the mouths. The mouths of the deep sea deserve their own extended attention, because they represent, perhaps more clearly than any other structure, the logic of what happens when food is rare enough, and encounters are unpredictable enough, that the opportunity cost of turning down a meal, any meal, regardless of size, regardless of whether the prey is manageable, becomes biologically unsustainable. In a world where you might not encounter another food source for days or weeks, the jaw that can only handle prey smaller than itself is a jaw that will sometimes fail when it matters most. So the deep ocean built jaws that impose no size restriction. And what resulted is a collection of mouths that, evaluated by surface standards, look grotesque, impractical, structurally unsound, and are, in context, the most efficient feeding architecture those animals could possibly carry. The fangtooth, Anoplogaster cornuta, is about 15 centimeters long as an adult, which is not a particularly imposing size for a deep-sea fish. Its teeth are the longest, proportional to body size, of any fish in the ocean. The lower canines specifically are so long that closing the jaw is a mechanical problem. The teeth are too long to fit inside the closed mouth, without penetrating the brain cavity. The skull solved this by growing dedicated sockets in its roof, pits that receive the tips of the lower canines when the mouth closes, allowing the teeth to slide up into the head without causing injury. The fangtooth bites, and its own teeth disappear into its skull. When the mouth opens again, they re-emerge, unfolding from the sockets. Those teeth are curved inward, so that anything caught in them is held against any force applied in any outward direction. The more a prey animal struggles, the more deeply it's drawn in. A 15-centimeter animal that encountered prey of almost any size could not only seize it, but hold it through sustained resistance until the prey exhausted itself. The teeth are not dramatic. They are economical. In a world where meals don't come often, what the fangtooth built ensures that no meal, once encountered, is lost. The viperfish, Chaliotis sloani, addresses the same problem from a slightly different structural angle. Its fangs are long enough that they extend outside the closed mouth entirely, curving back toward the eyes in a configuration that looks, in close photographs, as though the fish assembled itself from the wrong parts. In the dark of the midwater, the viperfish uses bioluminescent photophores along its body and on the tip of a modified dorsal spine, which functions as a lure to attract prey and then accelerates toward the attracted animal at speeds that generate sufficient impact force to stun it before the jaws engage. The skull is hinged to retract backward during the strike, allowing the jaws to open to an angle that makes the effective mouth opening wider than the head diameter, a neck joint that dislocates on purpose to allow the mouth to become briefly larger than the skull around it. The fish uses its own skeletal flexibility as an expansion mechanism during feeding. The black swallower, Chiasmodon niger, solves the problem from the stomach side rather than the jaws. Its jaws are unremarkable. Its stomach is not an organ with fixed dimensions. It is a membrane capable of expanding to accommodate prey, up to twice the fish's own length, and several times its body mass. When a black swallower encounters prey it cannot refuse, it swallows it whole, and the body deforms around the meal, the transparent skin stretching until the internal contents are visible through the body wall. There are preserved specimens in collections, recovered from the surface because the fish died and became buoyant enough to float up, which tells you something about how those encounters sometimes end, with prey items so large inside them that decomposition of the meal began before digestion was complete. The gases produced making the fish buoyant. The stomach that was built to handle anything encountered at depth became, in those cases, lethal. The fish ate something it could not finish quickly enough. The prey began to rot inside it, and the resulting gas carried it to an environment it couldn't survive in. The adaptation that was supposed to ensure no meal was wasted occasionally caused death by an excess of ambition. The gulper eel, Europharynx pelicanoides, takes the concept further than any other fish and turns it into a complete body plan. The jaw is enormous, loosely hinged to open to nearly 180 degrees, essentially a bag on a hinge. The body behind the jaw is a long, tapering tail with a bioluminescent tip used as a lure. The gulper eel doesn't bite its prey. It doesn't grab it. It opens the jaw to maximum extent and surges forward, engulfing prey and a large volume of water simultaneously, then drains the water through the gill slits while retaining the prey inside. The ratio of jaw to body makes the animal look, in photographs, like something that was assembled with the wrong scale of parts, the mouth accounting for a significant fraction of the total body length, the body behind it tapering to almost nothing by comparison. In the water, this design is devastatingly functional, against prey that has no warning of approach, because the eel produces no turbulence until the jaw begins to open, and by then the water is already moving toward the mouth. The gulper eel simply becomes, for a moment, a very large space that the prey is inside. Every night, one of the largest coordinated animal migrations on Earth happens in complete silence, in complete darkness, and has almost certainly been happening continuously for millions of years without any human observer. It is called the diel vertical migration, and it is driven by a simple arithmetic of survival. During the day, staying deep is safe, because the predators that hunt by vision have access to surface light, and anything shallow enough to be visible is at risk. When darkness falls, the benefit of that depth advantage disappears, because vision-based predators lose their advantage in darkness, while the food, the phytoplankton and zooplankton concentrated in the upper ocean, where sunlight supports their growth, remains near the surface. So every night, billions of individual organisms, lanternfish, krill, copepods, myctophids, small squid, shrimp, ascend from the depths where they spent the daylight hours. They rise hundreds of meters through the water column, feed at the surface or near it, and before dawn descend again to the safety of depth. The scale of this migration produces what sonar operators during World War II called the deep scattering layer, a false bottom that appeared on sonar screens at depths between 200 and 700 meters, and seemed to rise and fall with the cycle of day and night. For years, the Navy believed this phenomenon was a geological feature, some kind of reflective seabed that moved. It was the bodies of billions of animals moving together. The sonar was bouncing off flesh, the entire migrating biomass, dense enough to scatter sound waves, rising each evening and descending each morning, in a coordinated rhythm driven by nothing more than the logic of light and predation. Lanternfish alone, one of the most abundant vertebrates on Earth by total number of individuals, despite being almost entirely unknown to people who don't study marine biology, may migrate this way across hundreds of millions of individuals every single night in the Pacific. The total biomass involved in the diel vertical migration globally is estimated in the hundreds of millions of tons. It happens every night. It has been happening every night for far longer than our species has been observing it, and it will almost certainly continue happening every night long after we stop. Your breathing, in this moment, is doing something similar, a slow rhythm driven by chemistry and pressure differentials in your lungs, regulated without your conscious involvement, cycling in and out with the same ancient biological logic as those billions of animals rising and falling through the water column. Neither you nor the lanternfish is aware of being part of a pattern. Both of you are the pattern anyway. The siphonophore requires a pause, because it forces a question about what the word individual actually means. And the answer the deep sea offers is not the answer your intuition reaches for. A siphonophore is a colonial organism, but describing it as colonial undersells the integration. The colony is composed of zooids, genetically identical units, each one descended from the same original fertilized egg, through a process of clonal budding, each one specialized to perform a single function within the colony. Some zooids are nectophores, swimming bells that provide propulsion by jet. Some are dactylozoids, armed with stinging tentacles for prey capture. Some are gastrozoids, digestive units that process the captured prey and distribute nutrients to the rest of the colony. Some are gonozoids, reproductive units producing gametes. None of these can function independently. A nectophore removed from the colony cannot feed itself. A gastrozoid cannot move. A gonozoid cannot generate forward motion. Each one is a specialist that only makes sense in the context of the whole, and the whole functions as a single coordinated predatory organism. Even though it has no single brain, no central command structure, only chemical signaling between zooids. Praia dubia is the largest siphonophore known. In 2005, a specimen was filmed at approximately 40 meters in length, longer than a blue whale. longer than the longest known fish, longer than the longest documented individual organism of any kind in the ocean. 40 meters of coordinated zooids, trailing millions of stinging tentacles over an area of water that no single-bodied predator could cover, hunting by creating a passive mesh of stinging filaments through which prey cannot swim without contact. The siphonophore doesn't chase, it doesn't pursue. It extends itself through the water and waits for the ocean to bring food to the surface of its tentacles. And across 40 meters of coverage, the ocean obliges with sufficient frequency to sustain this structure indefinitely. If you tried to define whether Praia dubia is one organism, or 40 meters of distinct organisms acting in concert, you would discover fairly quickly that the question has no clean answer, and that the deep ocean is full of questions like this. Questions where the categories we use to organize our understanding of life turn out to be surface world conventions that the deep sea simply didn't adopt. The pressure in the deep ocean doesn't just change the mechanics of living things, it changes their chemistry. At Hadal depths, below 6,000 meters, the compression of seawater reaches values that would denature most of the proteins that surface biology depends on. The folded, three-dimensional structure of a protein is maintained by weak chemical interactions, hydrogen bonds, electrostatic forces, hydrophobic effects, and under sufficient pressure, those interactions are disrupted, and the protein unfolds, losing its shape and therefore its function. Most of the enzymes, structural proteins, and molecular machines running inside a surface animal would, at 400 atmospheres, stop working. The animal would stop working with them. Deep sea animals, particularly Hadal species, avoid this through two primary mechanisms. The first is trimethylamine oxide, TMAO, a small organic molecule that acts as a pressure-protective osmolyte. It stabilizes folded protein structures through a mechanism that researchers don't fully understand yet, but the effect is measurable. Proteins that would unfold at high pressure remain functional in the presence of TMAO, and crucially, the concentration of TMAO in deep-sea fish tissue scales with depth. The deeper the fish lives, the higher the TMAO concentration in its muscles. The Mariana snailfish, living at 8,000 meters, has among the highest TMAO concentrations ever measured in a fish. It is, in a sense, pre-treated against the pressure it lives in, its own flesh acting as a buffer between its proteins and the physics of depth. The second mechanism is the modification of the proteins themselves. Deep-sea enzymes have amino acid sequences, slightly different from their shallow water counterparts. Not different enough to change their function, but different enough to make them more resistant to pressure-induced unfolding. These differences accumulated over millions of years of selection at depth, each generation retaining the sequence variants that stayed functional under pressure, until the entire biochemical machinery of these animals was recalibrated for an environment that would destroy their ancestors. The Greenland shark's toxic flesh, saturated with TMAO at concentrations high enough to act as a neurotoxin in mammals, is a direct consequence of this pressure adaptation chemistry. The same compound that keeps the shark's proteins intact at depth makes the shark dangerous to eat. Its protection is our hazard. The Greenland shark itself is worth more time than it usually gets. Somniosis microcephalus grows at approximately 1 centimeter per year. It reaches sexual maturity at around 150 years of age, meaning every Greenland shark that is currently reproducing has been alive since the 19th century at minimum. The oldest specimens that have been aged using radiocarbon dating of the eye lens, which forms in the embryo from proteins laid down before birth and never replaced, acting as a biological time capsule of carbon isotope ratios, have been estimated at close to 500 years. Born in the early 1500s, alive today, drifting through the North Atlantic at 1 kilometer per hour, slower than almost any other predator in the ocean with a skeleton. Its primary hunting strategy for the seals it is known to eat is believed to be approaching sleeping animals. Seals rest at the surface and occasionally at shallow depth, and a predator moving at 1 kilometer per hour generates almost no pressure wave detectable by the seal's lateral line or other mechanosensory systems. The Greenland shark is not fast. It is patient in a way that makes patience sound like an inadequate word. 500 years of 1 centimeter of growth per year, of approaching sleeping prey too slowly to wake them, of surviving in waters cold enough to send any unprotected human into hypothermia in minutes, sustained by a biochemistry so specialized for that cold, dark, deep-pressure world that its flesh is poisonous in a warm kitchen. There are Greenland sharks alive today that were already adults when Galileo was building his first telescope. When Isaac Newton was formulating the laws of motion, a Greenland shark somewhere in the North Atlantic was already well into its 2nd century. The things that seemed important in human history across those 500 years, the things that filled books and caused wars, and reorganized the surface of societies, were happening in the same physical world as this animal, in the same decades, on the same planet, with the shark entirely uninvolved, drifting at depth at 1 kilometer per hour, alive and slowly growing, and completely indifferent to all of it. Let your shoulders drop. Let your jaw release whatever it's holding. Let the weight behind your eyes, the accumulated visual effort of the day, all those hours of focusing and processing and deciding, just go slack. The room you're in is still. The temperature is whatever your body finds easy to maintain without effort. Outside whatever window is nearest to you, the air is dark, and the world on land is either sleeping or moving through the ordinary business of being surface-bound and warm-blooded, in a world full of light and oxygen, and the comfortable, familiar physics of 1 atmosphere of pressure. Below every ocean on Earth, simultaneously, something very different is happening. The daily migration is playing out. Billions of animals ascending in the Atlantic, descending in the Pacific, ascending in the Indian Ocean, in a global biological tide, driven by the same sun that lit your day. The dragonfish's red searchlight is sweeping through water that no other animal in the vicinity knows is illuminated. The glass squid is hovering vertically, managing the opacity of its own gut. The 40-meter siphonophore is drifting, with its stinging mesh deployed. The Greenland shark is approaching something sleeping at 1 kilometer per hour. The barrel-eyes' green eyes are rotating inside their transparent dome, tracking shadows against the faint glow of surface light. 400 meters above it, and none of them know you exist. None of them are aware of the surface world at all. They are as complete in their environment as you are in yours, as embedded in the conditions of where they are, as you are in the warmth and the dark, and the familiar weight of gravity that your bones were built to handle. That completeness, that total, ancient, unself-conscious adaptation to a world you will never visit, is one of the things that makes the deep ocean so effective, as a companion for the edge of sleep. There is no urgency in it. There is no incompleteness. Every animal down there is already exactly where evolution placed it, doing exactly what millions of years of selection shaped it to do, in water that has been running this particular version of life for longer than the continents have been in their current positions. You don't need to go there. You don't need to understand all of it. You only need to know it exists, happening right now, below you, in the dark and the cold and the quiet. And let that knowing be what it is, an immense, ancient, ongoing thing, running without your attention, needing nothing from you, completely at peace in conditions that your body was never meant to occupy. The deep ocean will continue without you, quiet, ancient, and completely awake beneath the dark.